Hello, friends. In the corona pandemic period, we have uh, came in contact with the words such as a RT-PCR technique, in which the doctor or the technician, were taking our swab, and then the swab was tested by certain test, and then the result was came that the patient is whether positive or the negative, right? So we are going to study one of the very interesting technique which is used in the forensic lab and as well as in the virology or the bacteriological testing lab. So, hello friends, this is Miss D.A. Marvekar, working as assistant professor in the Department of Zoology, Rajeshri Chhatrapati Shahu College, Kolhapur. And today we will learn about the polymerase chain reaction. If I will ask you that at the murder case, I have with my one of my sample which is a suspicious one, and that is a very small in the quantity, and we have already studied that I will use the fingerprinting technique, I will use the southern blotting technique, but for using that technique or for the detection purpose I will require the large copy of the DNA, and it is not possible to extract the number of DNAs from that such a microliter quantity of the sample, right? So, if is it possible to, if you are having only one DNA molecule and you can convert the number of a molecule or the millions of a copy, so definitely this question will be answerable as a yes. And this technique is credited to this personality, that is a Carey Mullis, and he invented this reaction, that is a polymerase chain reaction, in 1983. He was working at that time as a CITES corporation in Emerville, California, and he received the Nobel Prize for this groundbreaking invention in the 1993. That is, after invention, the scientific community or the Nobel Prize distributing community uh, contributed or given the Nobel Prize to this scientist in after 10 years of the wait. So he also received a $10,000 from his employer status, and Bullet later also sold this patent to the Hoffman La Roche. Uh, it is not now we are going to discuss about this one, but definitely the credit goes to this scientist, a very famous scientist, that is Carey Mullis. Now, what happens when the DNA will get duplicated inside the cell? We know that whenever the cell will get divided, whatever the DNA contained in that cell, that DNA has to be get replicated or has to be get duplicated, so that when the cell will get converted into the two daughter cell, the two daughter cell will access or it will acquire whatever genetic material their parent cell was having. So therefore, during cell division, in the S phase of the mitotic period, the double-stranded DNA will get separated into the single-stranded template, and then the information on the single strand is utilized with the help of a primer and the DNA polymerase enzyme, and then after polymerization process, we will get two copies of the daughter DNA which are formed from the parental DNA and which are exact replica of this parental sequence. And so this is the general process of the replication which happens in our cell in day-to-day life during the whenever the cell is dividing. So if is it possible to carry out the same duplication reaction in the tube? Then the answer is a yes. And that technique is the polymerase chain reaction. This is the simple thermocycler machine in which here we are keeping our PCR tubes for the reaction of the PCR, and this is the display through which we can observe number of cycles which are occurring inside that machine, and by these keys we can handle or we can operate this machine, that how much type of how much cycles we required for the polymerization of the DNA. Now, when we are carrying out the polymerase chain reaction in the tube with the help of this thermocycler, we have to carry out the three important steps, that is denaturation, annealing and extension. Now, uh, This was the serendipity experiment, we can say that, because when the Carey Mullis was driving his Honda Civic on the Highway 128 from the San Francisco Road to Mendocino, he made that intellectual leap and he thought that he can design such a type of a machine in which he can duplicate the copies of the DNA. And so he invented this technique, and by using this technique we can amplify the DNA with the help of three steps in per cycle. Now if we are requiring the duplication of a DNA, in case of a cell, what will happen? The helicase enzyme will separate the double stranded DNA into the single strand one. In case of a cell, the enzyme which will synthesize the duplication of a DNA process, that is the polymerization of a DNA, that enzyme is DNA polymerase. For polymerization process, the startup will be generated by the primer, and that primer is synthesized with the help of a primase enzyme. And the trustability or fidelity of this process is very high, so no any wrong bases will be added during the duplication of a DNA in vivo, in the cellular type of a replication. But when I am repeating this process in vitro in the tube with the help of this technique, that is a PCR. So similarly, I will use each and every parameter in vitro, but that there is an alternative one. So in the PCR, instead of a helicase what it will do, it will complete the double stranded DNA into the single strands of the DNA. So here also in the PCR, by using the heat, double stranded DNA will get separated into the single strand. In case of a cell, we require the DNA polymerase enzyme. In case of a PCR, we require the Taq polymerase enzyme, which is separated from the special type of a bacteria which are present at the hot water spring type of an environment. In case of a primase, here we are using the different types of a primer, that is a forward and the reverse primer, and the fidelity of this process is less as compared to the cellular replication of the DNA. Now this is the picture which shows the how the hot water springs are there. So similarly, such type of a sources are used to isolate the bacteria which have adapted in such type of an environment, because here the temperature will be the very high above the 90 degrees centigrade, and the bacteria which are living in such type of an environment that have adapted to this environment. So whatever enzymes are secreted inside that bacterial cell are also acclimatized for this temperature. So obviously, if we are using the DNA polymerase from our cell for this machine, so it will not carry out number of steps. After first step, it will get denatured because it is a protein, and protein when we are increasing the temperature, it will get denatured. But here, if we are using the enzymes which is isolated from the bacteria which are growing in such type of an environment, then their temperature will be the optimum temperature will be the very high as compared to our normal DNA polymerase enzyme. So here we are using the Taq DNA polymerase, T for the thermus and X for the aquaticus. So the Taq DNA polymerase is extracted from the bacteria which grows on the harsh environment, that is hot water spring, and that bacteria is thermus aquaticus, right? So as all enzymes are a protein, but there are few who are exceptions. But here also this enzyme is a protein, and after optimum temperature, it will get crossed, it will get denatured, and therefore we are not using the DNA polymerase from our cell, but we are using the DNA polymerase from the Thermus aquaticus type of a bacteria. So in the PCR tube, what we will require, the requirement is the DNA sample of which we are interested to get the billions or the number of copies. We have to maintain the special environment in which this reaction will take place. So I will use the buffer. Then during replication in the cell also it will require the nucleotide triphosphate. So here also we will require the deoxynucleotide triphosphate in the simple form. We require forward primer and the reverse primer. and we require the Taq DNA polymerase enzyme. I will mix this mixture, I will close the lid and I will keep this lid into the thermocycler machine. I will set the protocol by the this step, denaturation, annealing and the extension. So in the denaturation in the machine, what will happen? As the denaturation temperature, that is 90 to 95 degrees centigrade, will reach, at that temperature the hydrogen bonds in the nitrogen bases in the DNA will get breakdown, and this double stranded DNA is converted into the single strands one. After single strand conversion, the primers, which are oligonucleotide chain, which are artificially synthesized, that primer are complementary to one of the end of the DNA template. So during annealing temperature, the primers will bind to the complementary region. And during extension, the DNA polymerase will bind and it will extend the end of a primer. So after completion of these three steps, it will complete one cycle. And so after completion of a one cycle, I will get two DNA copies from the one DNA. In the next cycle, these three steps will get repeated. So from this two DNA, I will get the four DNA. In the next step, again the cycle will repeated, and from this 4 DNA I will get the double of the 4, that is a 8 DNA copies, and so the cycle will continue. So we can adjust to the machine how much amount of sample we require, or how many cycles we are going to um, play, or we are going to adjust or going to set by the thermocycler machine. So for the denaturation, the temperature will be the 94 to 96 degrees centigrade. For the annealing, the temperature will be the approximately 68, and for the elongation process, it will require approximately 70 to 72 degrees centigrade. And so this cycle will complete within 1 to 2 minutes, and it is not feasible to uh, keep such type of a system so that it can easily decrease its temperature within a half minute, and it will again increase the temperature within a half minute. And so in case of a thermocycler, it will work on the Peltier effect, that is rapid heating and rapid cooling metals are used in the thermocycler, and due to which it is suitable for us to adjust the temperature, or we can vary the temperature in a fraction of a second, and it can be adjustable according to the time which is required for the different types of steps, that is denaturation, annealing and the extension. So here the during denaturation, this double stranded DNA will get separated at the 94 degrees centigrade. During annealing, the primer will bind, and during extension we will get the two. This is the denaturation step at 94. This is the annealing where the primer will bind, and then the extension where the DNA polymerase will extend the tail of a primer to get the two copies of the DNA. So this is the very nice representation of the amplification of the DNA, or when we are getting the duplication of the DNA. So we have already studied about this type of a reaction mixture. Uh, in the conventional method, uh, after addition of all this mixture in the tube, it was layered with the mineral oil so that it can prevent the evaporation of the sample. But nowadays, the lead of the thermocycler has modified in such a way that there will be no requirement of the mineral oil. So, such types of a cycles I can complete if I am having only one DNA molecule. Then I can get two DNA molecule in the one cycle. I can get the four DNA molecules, DNA molecules or the DNA strand in the two cycle, and so on. So, whatever the quantity of the DNA is required, accordingly I can adjust my cycle as a 20 cycle or the 30 cycle or the 40 cycle. Now there are different types of a PCR are there, such as a hot start PCR, touchdown PCR is there, RT-PCR, which was used for the detection of a COVID patient. And so there are different applications of a PCR. You can use this PCR for the amplification of a DNA if we are getting the sample in very small quantity. At each and every step we have to proceed that step after amplification of a DNA. In the archaeology, suppose you are with the one skeleton and from that you have extracted the DNA. But if it is in the very small quantity, then we have to amplify that before proceeding to study with the help of a PCR. In case of a epidemiological study also, we can diagnose the diseases caused by the bacteria or the virus with the help of a PCR. We can also diagnose the prenatal diseases with the help of this PCR. We can also study the mutation in the bacterial cell with the help of this PCR. We can also use this technique for the discovery of various drugs. We can study the we can use this technique in the fingerprinting, DNA fingerprinting. We can detect the unknown pathogen which is causing the disease one. We can use this for the sequencing, which was also used for the Human Genome Project. And there are lot of applications are there for this PCR. So I hope that you have understand this. Thank you so much.